Alright, hi guys, good afternoon. Today we are going to do the corrections for this paper, which is the October-November 2020. Okay, so first let's look at section A. This is an answer all questions in this section. And for this question, we have to pretty much just write down the answers however they kind of ask us what to do. So let's look at the first question. The flowchart shows stages in the life cycle of a flowering plant, which is one of our learning objectives. I think this one should be in topic 16, which is the sexual reproduction in plants. So we have stage 1 pollination, and then pollen tube formation and fertilization. And then after that you have fruit and seed formation, then fruit and seed dispersal. Then after that it's a blank, and then plant growth and flower formation. So before the plant grows, and we have seed here, so the seed has to grow first, and so this answer would be germination. So germination is basically when seeds grow into young plants. The name of stage 5 in the life cycle has not been included. During this stage, a radicle and a plumule are formed from a seed. Yeah, so radicle and plumule, correct place in the flowchart, state the name. So sorry, I should have read the question first, and that's what you should do, and then you answer, but we do get 1 point for writing germination there. Describe the role of enzymes in this stage of the plant's life cycle. So we're talking about germination here. And you know that germination is when the radicle and the plumule are formed. But how do they get this from? So basically, we should know the fact that the structure of the seed, you have it. Usually, we look at this kind of seed that looks like a bean, and on the outside. So what I'm drawing here, this one is the seed coat. And there's actually a little hole here. It's called the micropyle, something like that. Let me just have a quick Google. Micropyle. Micropyle. That's right. Let me see. Micropyle. What about the seed? Yep, it is called the micropyle. So that is an opening. Yeah, it is a hole in the seed coat that allows pollen tube. Yes. Is micropyle. Yeah, okay. This is where water actually enters. So micropyle is where water enters the seed. And why water is important is because remember that water is the solvent for chemical reactions, and this is where enzymes are important. Okay, so we know that this is a micropyle, and then you have the cotyledon here. So the cotyledon here contains the stored food, for example, starch. And then you also should have like a cute little leaf and also a cute little root. And that cute little leaf is called a plumule, and the little baby root is called the radicle. But clearly here the seed has not grown yet, radicle. So we want to grow these two parts right here. Go ahead and do. Okay. Okay, we do want to grow the plumule and the radicle. And to do that, that is the cotyledon's job. Okay, so what happens? So firstly, water will enter the seed, and remember that you need WOW factors to grow, so that's water, oxygen, and warmth or suitable temperature. But here we want to focus on mainly enzymes, so we can say that enzymes, sorry, handwriting. Okay, I'm just going to mute. So the enzymes that are found, there is quite a lot. They're pretty similar to the enzymes that we have. They have amylase, maltase, and lipase, and other digestive enzymes. So here we have 3 marks. So we can just give one example of an enzyme and talk about the substrate and what is its main use. I want to talk about amylase here because starch is also found inside the cotyledon. But maltose is not used for respiration yet, so I'm going to continue that. So my 4th point. And the 5th point here, so I'm just going to erase that. I'm going to talk about the use. And respiration is used for the growth of that radicle and plumule, because obviously they do need energy for these cells to pretty much divide and grow. So here we do have the 4th mark is talking about the usage. So I'm going to show you now in a different colored pen. So I'll use a blue colored pen. So although these are the numbers, I want to show you guys also the main points here. So saying breaking down will give you 1 mark. Saying the name of an enzyme, so you can use you can get a mark for amylase or maltase. Remember that only one enzyme is being given a mark here because it's only a 3-mark question, but it is like the requirement for this question because it says role here of enzymes and how it is important in this stage of life. So they do want kind of like a few different points. So the last, not the last one, but you do need to say what does it break it down into. So you need to talk about the substrate, and substrate here is starch, and maltose is a product. And then saying that it's used for respiration is also a mark. So if you mention any of these marks, then you'll get the point. If you do mention something about like maybe protease or lipase, for maybe protease is to break down protein into polypeptide chains, and or protease is to break down polypeptide chains into amino acids, and amino acids is used for protein synthesis, which is important because you know that all cells, especially plant cells as well, they have a cell membrane, and cell membranes are made up of protein, and a lot of the organelles are made out of protein. So that is pretty important as well in this stage of this germination part, because they want to focus on growth. Okay, part B, two of the stages in the life cycle of this flowering plant may depend on the action of an animal. Okay, identify two of these stages, and for each stage, describe how the action of an animal may be required. So, thinking about animal here, and this is it says here that it's a flowering plant. And so, actually, I think you would know this very well that pollination, especially in a flowering plant, would require animals. And the second part is actually seed dispersal. It's either seed or fruit dispersal because it depends, like, obviously the seed is found in the fruit and the fruit is being carried by animals. Okay, so the first stage we're talking about here is not germination, it is pollination. Okay, pollination. Okay, here, it says like which stage, right? So you can say stage 1, you can say stage 4, but if you want to be even more precise, you can just say pollination and food and seed dispersal, but the space is kind of small. That's why I'm just like, why is this so small? So I think I'm just gonna write down 1 instead here. So how do animals help in pollination? So they say that the pollen grains, they are sticky and they stick onto the body of the animal, and the animal will, when the animal is looking for the nectar, the pollen grain will then fall onto the stigma of the flower or a different flower. So basically talking about how pollination occurs. So we're just gonna say how do you know it has a rough surface? Because we're talking about animal pollinated plants here. Okay, what are the marks that you get awarded if yours is slightly different? We need to find out the fact that you wrote this part will be 1 mark. And then saying pollen grains and then talking about sticks or attaches onto the body of the animal, that's 1 mark, or even says like it moves onto the body of the animal, that's 1 mark. And then from anther, okay, sorry, I should have written down from anther to stigma here, so I missed that out. Moves to transfer the pollen from the anther, so that is important apparently, otherwise we don't get the mark. From the anther to the stigma together is actually 1 mark, so one without the other we won't get it. So on the same flower, different flowers would be 1 mark. So I'm guessing the whole definition of pollination has to have that from the anther to the stigma is very, very important. Okay, stage, which other stage? So we talked about the fruit dispersal, so that's stage 4. So we can write down number 4 here. And the act of the animal, so this is a different animal, okay? We different animals would do different things, like those insects like bees and butterflies, and even some small birds would help pollination, and they are very tiny because they're the ones that are looking for nectar. We don't look for nectar, but we do eat the fruits. So think about you as an animal that eats fruits. What do? You consume that food, and the seeds actually are undigested. They do have the seed coat, which is very, very strong, and it protects the seed. So once we ingest the seeds out, then those seeds will then go somewhere else. But think about a wild animal here, so maybe a monkey, or other sorts of mammals that do that. So this is the idea that we have. So what do animals do? Action of the animal. Oh no, I'm sorry, not which. This is that I wanted to like continue that sentence, but I don't feel like it anymore. So the animal. So we can say it like this. Okay, so where are our marks? So our marks come from this one here, which stage that you have mentioned. So that'll be stage 4. Okay, animal eating or consuming would be 1 mark. And then moving to a different area, because we're talking about dispersal here, so it has to be away from the parent plant. The seed is undigested. I just want to explain this because I wanted to kind of like connect that to egestion. Apparently you can also mention other things. So if you can say that the seeds are spat out or thrown away, you can also get 1 mark. Spat means to spit, but it is the past tense, so we say spat. Thrown away, because if you imagine like durian, you can't really swallow that. It's so big, like the seeds, so you throw it away. That is also considered as dispersal as long as it is away from the parent plant. Okay, question 2. In 1931, a scientist was pouring the powdered form of a chemical called PTC into a bottle. A small amount of powder accidentally blew into the air. A short time later, another scientist working in the same room said that she had a bitter taste in her mouth from the powder in the air. Describe with reference to the named components of the nervous system and the nervous pathway that led to the scientist detecting the powder tasting bitter. Okay, here we're seeing powder moving into the air, and so therefore moving to the nose, and then moving on to, so taste here should be, the receptor here is the taste buds, and the brain actually is the one that detects those changes. So when we talk about named components of the nervous system, we are supposed to talk about, the terms receptors, terms effectors, the term CNS, central nervous system, neuron. These are the brain, these are the components of the nervous system. So we're going to talk about the taste, right? So the receptor here would be the tongue. Okay, to CNS dot. I think that's pretty much it though, actually. I guess I can say that the CNS would be the one that actually will interpret the taste, actually. So let me just have a look. CNS function. Processing. Yeah, it coordinates information and sends an impulse to respond it. Okay. Let me just see if I can kind of close this in a better note. Carry. Okay. Okay, so I just want to show you guys this website that I like, GCSE. So what I would type is GCSE. But it says CNS, not CNA, CNS, nervous system. So here the CNS is the brain or the spinal cord. Peripherals are the nerve cells that carry information to the CNS. Okay, we have this. So it's a really good quick revision. Okay, receptors to effectors. So the thing is, like, your tongue doesn't really do anything unless you want it to, but you're just tasting it. That's what it kind of like ends here. So here we have receptors are specialized cells, so they detect the stimulus. And the stimulus here is the powder. So here the tongue detects the chemicals that are found in the powder and then sends it to the brain. So stimulus receptor sends sensory neuron to the relay neuron to the motor neuron. Effector responds. So here relay neuron brings it to the CNS actually to kind of like so they detect those changes as well. They interpret the information, but we are not giving a response in this idea here. So I just wanted to look at any other way that I can close off that information. Okay, I think I'm just going to say I'm gonna stop there because that would make a better sentence. Okay, so what are the marks that we need or that we will get from this? Okay, the first one is saying that impulse is first mark, receptor is your second mark, sensory neuron is your third mark, and the fourth mark here is talking about the brain. Is your fourth mark. Okay, the scientists pouring the powder did not detect any bitter taste. It was later found that inheritance of the two forms of the same gene, so that means allele, is determined if a person is able to detect the bitter taste of PTC or not. This is a really good fun fact. So sometimes, or not really sometimes, but you can, you have experienced this when someone says like, oh, this tastes sour, and you're like, no, it's not that bad. Everybody has their own unique genes for taste as well. That's why you might not like the same food as your friend. Okay, in this situation here, the scientist, one of them could taste it, the other one couldn't. Say the term used for different forms of the same gene. So I've wrote that down already, and that's alleles. Several years afterwards, it was found that environmental factors cause people to detect a range of different strengths of bitter taste from PTC. That means that not just the allele, but environmental factors play a part. This is a continuous type of variation where environment can have an effect. So, for example, height and weight. So this is continuous variation. So we have gotten these two are very straightforward. So we get one mark there, another mark there. Okay, chemicals similar to PTC with a bitter taste are produced by some plants to prevent them from being eaten. Animals that eat only plants have fewer genes that enable them to detect chemicals that taste bitter than animals that eat both plants and animals. Ooh, interesting. Animals that eat only plants have fewer genes. That means that animals that are herbivores will not detect bitterness of plants. However, omnivores that both eat plant and animals will have genes that can tell us that it tastes better. State the trophic level of plants in a food web. So here we're talking about plants, and they are the first trophic level. So in terms of trophic level, we can say either they are the first, but trophic levels, we also use the terms like producers, consumers, tertiary consumer, secondary consumer. So producer is a good word, but if you also say one, which is the or first, you can also get 1 mark from this. State the term used for animals that eat only plants. So we talked about this just now. It's herbivores. I'm gonna erase that because my handwriting looks really terrible here, so I'm gonna write that down again. Herbivores. Suggest why animals that eat only plants have fewer genes that enable them to detect chemicals that taste bitter than animals that eat both plants and animals. So we can talk about a few things here. We can talk about mutation, we can talk about natural selection. Another thing is that most of their energy comes from plants, right? Because animals that eat only plants would require to get most of their nutrients from mostly plants. So that gene of being less picky is being passed down from generation to generation, so helping them survive. So it is important for them to eat more of plants. Otherwise, if they're too picky, they would pretty much die off because they don't have enough nutrients. So how we would talk about this is all energy or nutrients. I'm sorry, I really don't like my handwriting. Okay, so that is the answer. Okay, so saying that all energy comes from the plants is 1 mark. Then saying that the plants will taste less bitter will be 1 mark. Saying that they will eat more variety is 1 mark. And then you can having this survival part will be 1 mark. So increasing their chances of survival or survival of the fittest is 1 mark. Although this is 2 marks, so if you mention any of these points, you can get your full 2 marks. Question 3. When an apple fruit is cut open, the cut surface of apple tissues quickly becomes brown and is less likely to be eaten. This change in color is shown in the diagram. White apple tissue immediately after being cut, and then afterwards they become brown. I think this is something that you've probably experienced if you have eaten apples before. So if you have a look over here, you have the first cut, and then 1 hour later, you can see that it becomes a bit more brown actually. You could experience this yourself as well. So it's not that it went bad, it just oxidized actually. So let's try not to be picky when we eat our apples. They're still fine. An hour later, it just became brown. The change of the color of the apple tissue is due to a series of chemical reactions. An enzyme called PPO acts as a catalyst for these reactions. So there's an enzyme that's found in the apple that causes it to become brown. What is meant by the term catalyst? Okay, catalyst itself, a catalyst is a chemical that speeds up chemical reactions and is not changed by the end of the reaction, and it can be reused. So we didn't talk about biological catalyst here, we're not talking about enzymes, but they just talked about catalysts. So we're just going to use the term without the protein part. So catalysts, substances, sorry, not chemicals. We use the term substances, I think. Substances. Okay, so the mark here is when you say that they speed up chemical reactions is 1 mark. Without being changed or any ideas about it can be reused would be 1 mark. If you want to use the chemistry definition, which is it lowers down activation energy, that'll also be 1 mark. I love using this as well because it really explains everything. Okay, the change of the color can be prevented by placing the cut surface of the apple tissue in boiling water for a short time. Ooh, you boiled an apple just to make it look pretty? Interesting. Explain this observation using lock and key hypothesis. Ooh, very fun question. So we are putting it in boiling water. We want to denature that enzyme, so we can pretty much talk about that part. So we can say here, so enzyme called PPO, so we can use the name as well. So anything to do with lock and key always has to be talking about the active site, and also about the shape of it, and talking about how okay, so the enzyme is shaped like lock and key. Specificity is important. what did it mention here? Okay. The change of color. Okay, so what are our marks? Where did they come from? So active site is 1 mark, denatured is 1 mark, or saying changes shape is 1 mark. Substrate no longer fits, or you can say that it's no longer complementary to the active site, is 1 mark. And then saying that no enzyme substrate complex is formed. This is important because like enzyme substrate complex is what the lock and key hypothesis is talking about. And then saying that the reaction or activity is reduced is 1 mark. Okay. Part B, scientists have recently used the process of genetic engineering to develop a type of apple that doesn't produce an enzyme PPO in its tissues. Ooh, genetic engineering. So just possible advantages of this type of apple. Okay. The good thing is the first part. The reason why they wanted to carry out this genetic engineering is that slower browning or no color change. Second is like more likely to be eaten, increases the profit. So anything to do with genetic engineering would have to do with increasing yield, increasing people wanting to buy those products. And the third one is to do with this whole process itself, where people can prevent it by boiling the apple tissue, so you don't need to boil it. So that would be really good. So it is kind of related to the previous question. So let's talk about this first, the one that is related to this question. Okay, so where are the marks for this? Okay, the first one. Okay, less browning is 1 mark. This part about it being more likely to be eaten or less likely to be wasted. So you can also say less likely to be wasted. It means that people don't throw it away because they think that it's gone bad. Increases profit is 1 mark, or increasing sales, and people no longer need to boil it is 1 mark. So we can say any of these 3 points. Okay, question 4. Hydroponics is a technique used to grow plants without soil. You can actually see this at the English block, the classroom at the bottom, just the first classroom. I don't remember which classroom number it was, but literally English block at the bottom, you can see like these tubes and these white tubes. I'm just going to show you if there is any in here. I'm sure there is. So if we just Google hydroponics, yep, it's like these white kind of like tube. Let me just move that here. But ours is like more circular and a bit more simple. So this is hydroponics. So what it means is that you don't use soil, you use water, and in that water you have like nutrients that are dissolved in it. So you have the light source at the top, you have nutrient solution here. So the nutrients are dissolved inside the water. Then you have the growing tray, that's where the plants are going to grow. We have a timer here, nutrients in the soil. You have air stone. So air stone, it's important because the roots also need oxygen. That's how they absorb nutrients, and that's how they absorb, mainly nutrients, because that is done using respiration. The roots still need to respire anyway as well. So you do have an air pump, nutrient solution pump, so timers. That means that they're not going to release all the nutrients at once. If there's too much, then you can get other things to grow inside the hydroponic solution. Imagine that there might be fungus in there and other bacteria in there, and if there's too much, then you can also get eutrophication. So here, let's look at the question now. The leaves of the plants are provided with a source of light for photosynthesis. State in either words or symbols the equation for photosynthesis. I prefer to use words, actually, because sometimes people forget to balance the equation. So let's do this in words first. So in words, it will be, so we need to start off with the raw materials, which is carbon dioxide plus oxy. Oh wait, no, sorry. Oh my god, what am I saying? Actually, there's a big space here. I'm just gonna see if I can fit this first. Plus water, and then arrow. Then we put sunlight. Oh my god, what am I writing? Sunlight. And then you make glucose plus oxygen. Okay, it actually fits. It's not too bad. If you want to write down the chemical equation, B6CO2 plus 6H2O, then arrow, sunlight. Usually we put in chlorophyll, if I'm not mistaken. Just going to write down C6H12O6 plus 6O2. Okay, so this is pretty straightforward, but let me see. The marks here apparently come from having the reactants part. So this part, let me see, this part to be one mark, and getting the arrows and the products right is your second mark. Okay, the roots of the plants are provided with a nutrient solution. The nutrient solution contains magnesium ions. So magnesium, we know that magnesium is needed for making chlorophyll, the green pigment that absorbs sunlight. Air is pumped through the nutrient solution using air stone that contains very, very small holes. Explain the advantages to the cells of the plant roots for pumping air through the nutrient solution using air stone. Okay, so we do have the part about these air stones. So we want air stones, and it says here that the air stone contains many small holes, and this anything to do with like little things and lots of little things, like very small things. So for example, you have lots and lots of alveoli, and then you break down food into smaller pieces. Everything has to do with increasing surface area. Okay, so we're going to talk about that first. Number one, increases surface area for oxygen to be released to Okay, so we have three main points, but there are 4 marks here that you can get from this. So saying increasing surface area is 1 mark. Oxygen is needed, so oxygen, and then talking about aerobic respiration together is 1 mark. And then saying that energy is needed for them to grow, and active transport to absorb magnesium is 1 mark. State the type of cell in plant roots that absorbs magnesium. Okay, this is called the root hair cell. So you're just going to write that down. Root hair cell. So that's 1 mark. Say the process that transports magnesium against a concentration gradient. Against a concentration gradient. So this is active transport. Just want to see. Okay, I don't know how long I've been talking. Actually, my back is hurting. Okay, part C. Farmers grew groups of 20 tomato plants of the same length of time in nutrient solutions with a range of different concentrations of magnesium ions. They determined the best concentration of magnesium ions for growth by measuring following for each group of 20 tomato plants. Okay, and then state the best concentration of magnesium ions is for growth, blank parts per million. So it's this one. Okay, so it says here the best concentration of magnesium ions for growth. So mean height, mean mass of plant stems and leaves. So we're looking at the ones that have quite a lot. And you can see here the largest for plant stem is here, and here as well. Stems and leaves, the height and then the mass is the largest, so it's clearly 125. Explain your answer, and then the knowledge of importance of magnesium ions to the plants. So the first part is to do with knowing what you can see from this data here. You can see that it shows the largest height and mass being produced, and then the second part is that you need to know about how, or mention the part about magnesium being needed to make chlorophyll, and chlorophyll is actually important for photosynthesis and so the production of starch and all that. So that is all you need to mention. So here we have the first main point, which is tomato plant. I want to use glucose. Okay, so where do we get our marks from? Saying that you have the largest here, and then magnesium meter for chlorophyll together is 1 mark, and then saying that you have more chlorophyll means more photosynthesis, or you can say increase rate of photosynthesis, and saying that it produces a lot of glucose is 1 mark. Okay, alright, we're not even in section B yet. Okay, question 5. Okay, the diagram shows the total volume of water entering and leaving the organs of the human alimentary canal during 1 day. Name the organs labeled A and B. So A is over here, this long part right here. B is this bag-like organ. Okay, we know what those are. So A is the esophagus. How do you spell it? O-E-O-So-Fa-gus. And B is your stomach, and you will get 2 points from that. Calculate the volume of X absorbed from the ileum. So you have X here. Okay, this is basic maths. So you have 3,500 must be equals to, so wait, what? Oh wait, what? Okay, hold on, sorry. Okay, total volume of water leaving 4,200 cm cube added by large intestine. 1,250 absorbed from the large intestine. Oh, absorbed in the ileum. So it's added, and then absorbed, and then added, then absorbed, and then lost. So it's basically something like this. 3,500 plus 1,500 plus 4,000 plus 200 so this is added and then equals to what is absorbed, so X plus 1,250 and then plus sorry, 1 to 50, and then plus what is lost during ingestion, 150. Okay, I need to get my calculator out. Right, so that's 3,500 plus 1,500 plus 4,000 plus 200. That's 9,200 equals 2. X plus 1,250 plus 150, 1,400. And you bring it to the other side. Or I don't know what you do nowadays. You minus here, right? And then you minus to the other side. Whatever your maths teacher taught you. But basically x equals to 9,200 minus 1,400. Yes, I got this right. It'll be equals to 7,800. What is the unit? cm cube. Okay, that was hard. Oh no, I should have written it down. I'm sorry, but that should have been written down, like down here, like the working should be written down there, but I'm a bit too lazy to bring it down now. Do you still want me to? Maybe I should. I'll show you how to write this down in that section. Okay, I'm just going to take a photo so that I remember what I wrote. Then I'm going to write it down there. Okay, so in and out, so like added and absorbed or lost. So absorbed or ingested lah. So maybe okay, so that would be, so you're gonna write down 3,500 plus 1,500 plus 4,000 plus 200 equals 2. Sorry, I forgot the cm cube. Okay, there you go. Alright, describe the absorption of water from the ileum. Okay, so this means whenever we're talking about absorption of water and describing it, we have to talk about the movement of molecules. Okay, so here, movement of molecules, and we're talking about water especially, would definitely be osmosis. You can also talk about diffusion. And so in terms of glucose, for example, glucose is actually an amino acids, especially glucose actually can be absorbed via active transport plus diffusion. Okay, water is not absorbed via active transport. We know this, water is always osmosis and diffusion, but glucose is very, very needed by our body for energy during respiration. So the body will do whatever it takes. It will even use up energy using active transport to absorb glucose. And when does this happen? Is only if the villus, the cells of the villus, has high concentration of glucose, but there's still a little bit, like on the outside, in the ileum. There's still a little bit, so they do still want to get that in. So that will be via active transport. But remember, we are talking about water here, and water is absorbed. Oh my goodness, sorry, dropped my iPad. Okay, water is absorbed into where water is transported, which is into the blood. So that is into the blood vessels that are found in the ileum. So if you remember, this is a really bad drawing of the ileum, but the ileum is like this. You have the lacteal. That's for the absorption of fat. And then you have like something like this. You draw it kind of like a net. And this is your blood vessels. Okay, so going in here and then going out. So going in, that would be an artery. This would be a vein, just away from that organ and towards the heart. And then you have on the outside, oh sorry, okay, yep, that's okay. On the outside here is like this layer of one cell thick cells. So we draw them kind of like this. And then it has nucleus in the middle here, and these are the epithelial cells of the epithelium of that villus. So this whole thing is a villus. Okay, I'm gonna stop talking now and show you guys the answer. So water. So it says here or diffusion, so maybe or diffusion or osmosis. So I'm sorry, I'm just going to use osmosis instead, because I want to use just one. So I want to use water potential gradient. If you want to talk about osmosis, diffusion, you could say water concentration gradient, but we always use osmosis. So let's talk about water potential gradient. Okay, just to check, but it says, and then I want to talk about the theory. Just wanted to look at the structure of it again. Yeah, lining here, but it doesn't really mention it specifically. What's the epithelial villi structure? Yeah, epithelial cell. Okay. Okay, where are our marks? So we have osmosis, one mark, or diffusion, you can say that one. Water potential gradient, but remember if it's diffusion it'll be water concentration gradient. and then into the villi or the epithelial cells of the villi. That'll be 1 mark, and then so these two are like considered the same mark, and then talking about moving into the blood and the capillaries is 1 mark. State how a named organ causes some of the water absorbed from the ileum to later become water in the urine. Ooh, which organ helps to form urine? And this is actually your kidney. Okay, so here we have the kidney. So it filters, kind of the water goes into the blood, right? So the kidney, what it does is that it filters the blood and removes excess water. So I just added the bladder part as like an extra, but it won't give me any marks, by the way, because the kidney is the one that gives you the mark. And then saying that it filters the blood and removes, so you have to say remove excess water is already 1 mark, so that's 2 marks done. Yay, finally, section B. Okay, the diagram shows a vertical section through the human skin of a hot environment. Okay, so this is the one that always confuses me. I'm like, is that pressure or is that temperature? This is the nerve ending. So the one that I always look at, I do have a book outside, so I'm just gonna go grab my book. Oopsie, dropped something. Okay, found my book. I think I have like printed this off for you guys. But so there are different types. Oh, okay, this one doesn't really show it actually. You have temperature receptors and you have the nerve endings. Okay, my book doesn't show it. I really need a book that shows me this. So let me just find a GCSE bite size if they have it. Skin. Would it be this one? It's actually the structure of the skin though that I actually want. One structure of the skin. Would be homeostasis. It is under homeostasis, but but see, it only shows, doesn't show everything. So wet glands. I want the full structure of the skin. Because there are different types of like of these nerves, and sometimes they do ask you that. So can I filter this to a large size so that it is clear? Let's see right here. Is that showing sweat glands, dermis? Nope. Oh, there you go. It's a good one. Okay, you have that, and you have here. But we're looking at this one, isn't it? Yeah. Pacinian corpuscle. You have thermoreceptor, that means temperature. And then you have here thermoreceptors as well, so it could be temperature as well. So let me just see Pacinian corpuscle. Pressure. So I would assume this one here is for pressure actually. Okay. My curiosity is complete, resolved. Okay, name and outline the role of P and Q though. Although it's not really mentioned here, but P and Q. So you can see here, you have actually this, if there's an arrow here, that'll be an artery. And it branches out, isn't it? It branches out here. So this should be an arteriole. Okay. Why is this not a capillary? Because capillaries are between tissues and you don't see any cells in between here. So P here is your sweat gland. So name and outline the role of P and Q, so you need to know the name of it. Obviously the sweat gland is to produce sweat, and it releases sweat through the duct. Sweat. Is that the duct? Yes. Oh, funnily enough, it does say capillary though. So difficult. Okay then, sorry, cancel what I said. This is a capillary. Brings blood to the surface. I guess because this is a capillary and it connects the artery to the vein. True, it is connected here, so I was wrong. This is actually a capillary. Okay, P is sweat gland. Outline the role of P and Q. Name and outline, okay. Okay, where are our marks? So we have sweat gland, which is your first mark. Produces sweat is one mark, and releases sweat is also one mark. So it's either or. And then talking about how sweat evaporates, and then cooling the body temperature is one mark. Okay, Q is a capillary. What does it say here? Ah, control of body temperature. Yeah, we have to mention body temperature. So it's already talking about in a hot environment here. Okay, so capillary is 1 mark. Then saying that blood flow is increased is 1 mark. And then bringing blood near to the surface is 1 mark. 1, 2, 3, 4, 5, 6. I'm missing something. Oh, removal of loss of heat. So here, cooling the body down. Okay, I think I need to mention that. Oh yeah, carrying heat away. So that would be 1 mark, but I think I want to say surface heat is lost on the surface of the skin. Okay, so the part about saying carrying heat away is already 1 mark. So, or saying that heat is lost, but that's only 1 mark if you mention any removal or loss of heat once in the answer. I explain how the process of temperature control in humans is an example of homeostasis. So remember, the definition of homeostasis is the maintenance of a constant internal environment. So we have to mention here that temperature is the one that is kept constant here. And what is negative feedback is when there's a change or an increase or decrease of that internal environment, and the factor here we're talking about is temperature. It is being corrected or reversed back to the normal temperature. Explain how the process. Okay, it's an example. So homeostasis. In homeostasis, okay, I'm just sorry. Can I use red pen here? So what we mean by internal is right inside our body. So what we mean here by is the internal body temperature increases or changes above normal. Okay, so saying that it is kept constant, and then saying internal environment is 1 mark. And then for negative feedback, you have to say there's a change. So you can say either increase above normal or decrease, or you can say just a general change, and then the body corrects. So it corrects this change. For example, lowering the body temperature. So that is 1 mark, giving all 3 marks. Okay, now we have excretion. So what is the definition of excretion? Excretion is the removal of toxic waste products. And you have to, or you can give an example. So kidneys are organs of excretion, and it removes urea, so I guess we can mention that. Okay, here they're very lenient, where they just give you saying the removal is 1 mark, toxic is 1 mark, and then waste products given an example is 1 mark. So 3 marks is done here. A person with kidney damage requires the process of dialysis to take place every several times each week. Okay, diagram shows a section through the internal structure of a kidney machine. Okay, so we have to the person's blood is here, dialysis fluid in, out. Blood of the person goes through this white tube here. Describe the structure of dialysis membrane and explain how it functions. Okay, so we need to mention about what we can see in this dialysis membrane. And the membrane is this thing, that line is a membrane. And you know that membranes are kind of like sheets of like, a membrane is kind of like a covering. So we're talking about this here. And this membrane is actually not solid. I mean, okay, it is solid, but it's not blocked. It actually has holes in between them, and they're small holes, so they are actually semi-permeable. And what helps is that for dialysis, what happens is that you know that your blood carries everything. Oops! Oh my gosh! Oh my gosh! Oh, thank God! Okay, let me just save everything. Okay, good. Okay, all right. So blood enters here, right? It enters here, and then so this is the blood that contains urea. Your blood has everything, urea, oxygen, and whatever, and it is entering this dialysis fluid. There's here, and here, and here. And what the thing is, the blood that we're looking at here that comes from the person, if the person doesn't have a working kidney, this blood will contain high in urea, high in toxins, high in carbon dioxide, and so the fact that urea is mainly removed when the kidney filters the blood, that is what we really want to remove from the blood actually. So urea and other ions that are in excess, and all the toxic waste products needs to be removed. So what happens is that, so I'm just going to draw green color for the toxins. So let's say that these are like urea and toxic waste products. Since there is a lot in the blood, what's going to happen, and less inside the dialysis fluid, so what happens is that this will actually diffuse into the dialysis fluid. So it's going to eventually move into the dialysis fluid, and then in the end the blood that would actually move back, so this one here following back, so here following through there, sorry, I was just really excited when I colored this. Let me just follow that train. So the blood that moves in here, so in this tube, and moving back into the person's body, will finally not have urea, because the urea has already gone into the dialysis fluid, because it has less, because of the concentration difference. So you're going to have blood with low urea return back to the body of the person, of that patient. Okay, so how do I explain this in sentences? Okay, I'm just going to write that down. even excess water actually. And that is quite important as well because we need to maintain the osmotic pressure as well. Okay, here's the part about it being folded is 1 mark, and then large surface area is 1 mark. Diffusion, okay, wait, sorry. Semi-permeable, or you can say partially permeable, is 1 mark. And then diffusing is, or osmosis in terms of water, is 1 mark. And saying that the waste products, so you can mention they are small though. Small waste products and toxin and water is 1 mark. And then saying that they pass out includes in that 1 mark. And the last part is about saying that large protein molecules and blood cells, they do not pass out, because that is important for the blood of the patient. You want to remain that, you don't want that to be gone. Explain why dialysis fluid in the kidney machine must be continuously replaced. So this is the similar thing that happens in your lungs, whereby in your lungs you have that alveolus, and the alveolar wall is made up of like semi-permeable membrane, and you also have capillaries that are flowing right next to the alveoli, and blood. So actually you have your blood in here, so that's your red blood cells, and blood actually passes down here continuously, and that is because of blood pressure that is pushing the blood away, and we need that movement, and that movement actually maintains a concentration gradient, and that is the important part. So number 1, so continuously being replaced, this maintains. Okay, so maintaining gradient, concentration gradient is 1 mark, preventing buildup is 1 mark, and then allowing diffusion to continue is 1 mark. So what we mean by this, if you guys don't get it, is the fact that, so you have kind of for diffusion, it's like 2 different areas. So you have your blood here, and then you have the membrane. So I'm just going to draw the membrane as like this, because it is pretty much partially permeable in it. Okay, and in the membrane itself, okay, so I'm just gonna draw blue for the gaseous fluid here. So you have these 2 areas here. Okay, so the blood is coming in this way, and then the gaseous fluid is coming out the other way. Okay, what happens is that you have toxins that are found that are coming in, and this is a lot. And because the gaseous fluid is moving out here, so what happens is that this will enter here now. So every time the toxins are moving in, that means that there is a buildup of toxins on the other side here of the gaseous fluid. So what you want to do is that when the next blood that comes in and brings in like toxin here, at this area, we want, so in this area, we want this to be in lower concentration than here compared to here. This one should always be in high concentration so that it will diffuse out. But in this case, if we don't remove the gaseous fluid here, then there's a buildup of that toxin, so we want to remove it. So that's why it's always flushed out. So you have this high versus low concentration, and diffusion will happen because diffusion is a movement of substances from higher concentration to lower concentration down a concentration gradient. Yay, section C. So this is an either or question, so you can either pick 1 or 2. I mean, like one question or the other one, not both. If you do answer both, the first question that you answered will be picked and the other one will not be marked, basically. Okay, so describe the principal roles of, in terms of coordinating and regulating bodily functions of the cerebrum. Okay, cerebrum, so let me just have a look at the brain. So here we have the cerebrum, which is this part, and that is to do with pretty much thinking, memory. We have hypothalamus, which is this part here, and actually the bottom here is the pituitary gland, and pituitary gland is the one that is a master gland and makes most of the hormones. Hypothalamus is the one that actually detects the changes in the blood, so that includes concentration of carbon dioxide, the body temperature as well. Cerebellum is this one that looks like a flower. It's quite pretty-ish, and it controls balance and coordination. And then you have the medulla oblongata, which is over here. What does the medulla do? Medulla controls the unconscious activities such as heart rate. So heart rate, breathing rate is this one. And then cerebellum here is to do with balance and bodily coordination. Okay, that is a little bit of a, okay, so here we have 3. Okay, so cerebrum. So you have to mention all of the main functions there. So number 1. Okay, so the points that we want is thoughts and thinking. Memory is one. You can also say intelligence or learning. So learning or intelligence. Emotions is one. So speech or language, you can say that as well because the mouth is there. Then other senses. So senses, for example, sight, hearing, taste, and smell. Those are just 1 mark. Voluntary action is 1 mark as well. Okay, cerebellum. You can mention this because apparently it wants to talk about instinct and subconscious or involuntary actions. So your 1 mark is this, and then involuntary action is 1 mark. Okay, the hypothalamus. You can just say homeostasis, honestly. Okay, and your marks comes from saying that it detects changes. It triggers a response. Okay, maintenance of constant internal environment or homeostasis is 1 mark. Detects the increase or decrease of temperature, and so, okay, increase or decrease is 1 mark. Temperature, concentration, and everything that is found in the plasma is 1 mark. Okay, the last question, question 9. Explain how surgery may be used as an effective method of birth control in a man and a woman. Okay, surgery here, we're talking about vasectomy. And we learned that this can be done both in the male and female. So birth control, obviously you want to control birth. You don't want to have fertilization happening here, so we don't want the sperm to meet the egg. So here when we have the vasectomy in a man, we are cutting the sperm duct, and in the woman we are cutting the oviduct so that the sperm doesn't reach the egg. So here in both of them, no fusion of gametes is happening. So here okay, so just so you know, semen is actually seminal fluid plus sperm. So me using the word semen here, I don't think that is the best, but it is in the answer scheme, so yeah, we can probably use that here. I can say in the fluid. So the sperm duct is cut is 1 mark, and then actually is cut is 1 mark, and then sperm duct is 1 mark, and then there's no sperm is 1 mark. So no sperm is released is no sperm is released as 1 mark in the semen. It has to be that. Okay, sorry. Okay, and the woman, the oviduct is cut or tied. Actually, they tie it up. Okay, wait, sorry. I'm just gonna continue the sentence. So that after intercourse, the sperm will not reach the egg. Okay, so over here the mark says, because we've already talked about cut, but here which part is the one that we're looking at? So oviduct. The egg does not reach the uterus. Don't know how that. Don't know how that is important, but apparently it is needed in the answer scheme. Okay, there's 5. 1, 2, 1, 2, 3, 4, 5. Okay. Surgical methods of birth control do not prevent the spread of pathogens. Okay, the reason being is because spread of pathogens is through the fluids. So the fluids here, the semen is still released, and the fluids that are found in the female, their female fluids are also being shared here with their partner in intercourse. So things like HIV can still be transferred. So describe methods that will prevent HIV. So what we want is that the fluids will not reach each other. So there is that use of physical barrier. So there are other reasons. So how this can be controlled, you can use antiviral drugs to treat it, because we want to prevent the spread of HIV. So this is methods to make sure that HIV is not spread from one person to another. I'm just going to write it down here. Antiretroviral, or just say drugs, can be used so that means that they're going to prevent the spread, they're going to stop HIV. And 2, remaining with one partner, so that if they have more than one partner, then they could spread it. Abstinence is also one, because if you don't carry out intercourse, you will not get HIV. Education about HIV and AIDS. So that means people will know what not to do. Using condoms as a physical barrier. So no fluids are being shared here. Testing for HIV to trace, contact tracing actually. So this means like people know that they have HIV so that they can not spread it. So the last one is sterilizing. Razors. So this is important when it comes to like razors and barbers. Okay, so all of these are pretty much 1 mark. Yeah, you can, for the part on remaining with one partner, you can say not prostitution, or no, not having sex or intercourse is also 1 mark as well. So that is the end of this paper. Let me know if you have any questions and text me if you need any help. Bye guys.